What if I told you the brain has messengers that actually send signals backward, or, get this, are made of gas, or even ones that double as the cell's own energy currency? It sounds pretty out there, right? In neuroscience, we've got a pretty clear rule book for how brain cells are supposed to communicate. But today, we're diving into the rebels, the atypical neurotransmitters that basically shred that rule book. And in doing so, they show us just how wonderfully bizarre and sophisticated our brain's conversations truly are.
So here's the game plan for this explainer. First, we're going to lay down the law by opening up that neurotransmitter rulebook. Then, we'll meet our lineup of incredible rule breakers. We've got cannabinoids that talk backward, and nitric oxide, a messenger that's literally made of thin air. We'll also look at neuropeptides and their unique origin story, melatonin, which kind of acts like a hormone, and finally, ATP, the energy molecule that's moonlighting as a messenger.
Okay, so to really appreciate how wild these renegades are, we first need to understand the rules they're breaking. Let's establish the gold standard criteria that define a classical or typical neurotransmitter.
Think of this as the 5-point checklist for any conventional brain messenger. First, it has to be synthesized, right there in the presynaptic terminal. That's the sending end of the neuron. Second, it's got to be stored in these tiny little bubbles called synaptic vesicles, all ready to go. Third, its release into the gap between neurons, the synaptic cleft, has to be triggered by an influx of calcium. Fourth, once it's out, it has to cross that gap and bind to specific receptors on the receiving postsynaptic neuron. And finally, and this is absolutely crucial, there has to be an off switch, a mechanism for its inactivation or removal to end the signal. Keep these 5 rules handy, because every single one of them is about to get bent, if not completely broken.
Our first rule breaker is the cannabinoid system. And the most radical thing about these molecules, they completely flip the script on the direction of communication. Instead of the signal flowing forward from the presynaptic to the postsynaptic neuron, they send signals backward.
Let's trace the signal on this diagram because it shows this backward talk perfectly. So everything kicks off in the postsynaptic neuron down at the bottom. When it gets really excited or depolarized, its voltage-gated calcium channels open up and calcium just floods in. This calcium rush awakens enzymes that instantly whip up endocannabinoids on demand, using lipid precursors that are already hanging out in the cell's membrane. Now here's the really cool part, because they're basically oils, they don't need a vesicle, they just slip right out of the cell, diffusing backward across the synapse. Their target? These CB1 receptors on the presynaptic terminal. When they bind here, it's like hitting the brakes. It inhibits calcium from entering the presynaptic side, which ultimately cuts down the release of its main neurotransmitters. It's a super elegant and precise feedback loop.
So, you see, this isn't just some quirky little exception. It's a fundamental feedback mechanism. Classical transmission is a one-way street. The presynaptic neuron talks, and the postsynaptic neuron listens. But with retrograde signaling, that listening neuron can talk back. It can literally tell the talking neuron to pipe down or not so loud. This makes endocannabinoids incredibly important modulators for fine-tuning brain activity, especially for our big workhorse systems like glutamate and GABA.
So let's meet the key players in this system. The 2 main endogenous ligands, the brain's own cannabinoids, are anandamide and 2-arachidonylglycerol, or 2-AG for short. Their primary targets are CB1 receptors, which are all over our neurons, and CB2 receptors, which are mainly on immune cells. Now, they're not identical. Anandamide is only a partial agonist at CB1 and doesn't do a whole lot at CB2. 2-AG, on the other hand, is considered the true agonist for those CB2 receptors. As we saw, they are made on demand from different lipid precursors, NAPE for anandamide and DAG for 2-AG. And to shut the signal off, they have their own personal cleanup crews. They get transported back into cells and are broken down by very specific enzymes. An enzyme called FAAH, that's fatty acetamide hydrolase, takes care of anandamide, while another one called MAGL, or monoacylglycerol lipase, handles 2-AG.
Now, you've almost definitely heard of THC, right? Tetrahydrocannabinol. This is an exocannabinoid from the cannabis plant that essentially hijacks this natural system. It binds to our CB1 receptors, and that's what causes its well-known psychoactive effects. But this hijacking also gives it some serious therapeutic potential. It's being looked at for treating things like the spasticity and multiple sclerosis and for chronic pain. It's also known to reduce intraocular pressure, making it a potential treatment for glaucoma.
All right, from a messenger that talks backward, we're now moving to one that breaks an even more fundamental rule of biology. Our next rule breaker isn't a solid or a liquid molecule at all, it's a gas. Meet nitric oxide, or NO, a highly reactive, super short-lived gas that somehow works as a crucial signaling molecule in the brain.
And this is the central puzzle of nitric oxide, isn't it? I mean, how can something as fleeting and uncontrollable as a puff of gas act as a precise messenger? You can't store it in vesicles, it's gone in seconds, and it passes through cell membranes like a ghost. Well, the secret isn't in storing it, but in making it at the exact moment and in the exact location it's needed for immediate action.
So here's the cause, mechanism, consequence pathway. It all starts with an enzyme called nitric oxide synthase, or NOS. NOS grabs the amino acid arginine, and with the help of a whole team of cofactors, we're talking FMN, NADPH, FAD, BH4, and calmodulin, it converts arginine into another amino acid, citrulline. And in that reaction, nitric oxide is produced as a byproduct. Because it's a lipid-soluble gas, NO isn't contained, it immediately diffuses out, acting on nearby cells. Its main target is another enzyme, guanylyl cyclase. Activating this enzyme ramps up the production of a second messenger called cGMP, which in turn activates protein kinase G. A key effect of this whole cascade is vascular relaxation, the relaxing of blood vessels. This is so important that NO actually used to be known as endothelium-derived relaxing factor, or EDRF, way back before we knew exactly what it was.
Now, the story gets even more specific, because there isn't just one NOS enzyme, there's a family of three. You've got endothelial NOS, or eNOS, found in the endothelial cells lining our blood vessels, where it regulates blood flow. Then there's neuronal NOS, or nNOS. It's in postsynaptic neurons and is activated by calcium. It's a huge player in synaptic plasticity and memory formation. And finally, there's inducible NOS, or iNOS. This one is typically in immune cells and can be induced or switched on during an immune response, where it plays a major, and sometimes damaging, role in neuroinflammation.
So nitric oxide is a perfect example of a double-edged sword in the brain. In controlled amounts, it's like a master architect. It regulates glutamate receptors to strengthen connections for learning and memory, a process we call long-term potentiation. It's a diplomat, facilitating communication between neurons and their astrocyte support cells to help get them energy. It even helps reinforce the physical structure of synapses. But when things go wrong, like during a stroke, the brain gets flooded with it. As a highly reactive free radical, it can team up with another radical, superoxide, to form a truly nasty molecule called peroxynitrite. And that stuff is like a wrecking ball for our cellular power plants, the mitochondria.
All right, our first two renegades broke the rules of direction and physical state. Our third class, the neuropeptides, they break the rule of location. See, their story doesn't start at the synapse. It starts way back in the cell's central command, the DNA in the nucleus.
Here's the fundamental difference. Think of a small transmitter, like dopamine, as being put together in a little local workshop, right there at the axon terminal, where it's needed. It's fast, it's efficient. Neuropeptides, though, are totally different. They're more like a complex machine built in the main factory, the cell body, or soma, using the full machinery of protein synthesis, and then they have to be shipped all the way down the long supply line of the axon.
Let's follow that journey. So a gene in the nucleus gets transcribed into messenger RNA. That mRNA is translated in the rough ER into a huge pre-pro peptide molecule. This then gets processed and snipped down into a smaller propeptide, which is then packaged into large, dense core vesicles. These vesicles get transported down the axon. Now, their release requires intense, high-frequency stimulation, and they could even be released from the sides of the axon, not just the tip. This is called volume neurotransmission. It's less like a targeted whisper to one neuron and more like a broad announcement to a whole neighborhood of cells. And once they're out, there's no going back. They're destroyed by enzymes in the cleft called endopeptidases. A fantastic example is the pre-pro-opia-melanocortin gene, which is like a molecular Swiss Army knife. It gets processed into multiple active peptides, including the pain-relieving beta-endorphin and the stress hormone ACH, all from one single blueprint.
There are tons of neuropeptides. You have somatostatin, which helps modulate cognitive and motor functions. Cholecystokinin, or CCK, is actually the most concentrated neuropeptide in the brain. And the tachykinins, like substance P, are heavily involved in transmitting pain signals. Let's focus on the opioids, which act on G-protein-coupled receptors, like the mu, delta, and gamma types. Here's exactly how an opioid like beta-endorphin blocks pain. When it binds to a mu receptor, it does two things at once. On the presynaptic side, it closes calcium channels to stop the pain-signaling neuron from releasing its transmitters. At the very same time on the postsynaptic side, it opens potassium channels. Potassium rushes out, which hyperpolarizes the neuron and makes it way harder to fire. It's a really powerful two-pronged attack on the pain signal.
Okay, our next atypical messenger really blurs the line between a hormone and a neurotransmitter. Let's talk about melatonin.
It's synthesized in the pineal gland, and it's best known as the hormone of darkness, our body's main timekeeper, regulating our sleep-wake cycles.
This slide just captures melatonin's daily rhythm perfectly. Over on the left, you can see the simple recipe. The neurotransmitter serotonin gets converted in 2 steps to melatonin. But the real magic is in the timing, which you can see in those graphs on the right. Just look at the key enzyme, ANAT, in that second graph. It's basically asleep during the day, but then its activity just skyrockets when the lights go out. This enzyme is the master switch that drives that nightly surge in melatonin that you see at the bottom. It's a direct, elegant link between the light-dark cycle, chemistry, and our internal clock.
So let's just quickly recap its life cycle. It's made from serotonin, packaged into vesicles by the VMAT transporter, and released in a calcium-dependent way. It primarily acts on 2 G-protein-coupled receptors, MT1 and MT2, located in the brain's master clock, the suprachiasmatic nucleus, or SCN. MT1 receptors help regulate the circadian rhythm itself, while MT2 receptors are involved in phase-shifting that rhythm. But melatonin also has a secret weapon. It has a non-receptor job as a powerful antioxidant. Because it's amphipathic, meaning it's comfortable in both fatty and watery environments, it can easily slip through cell membranes and neutralize damaging free radicals directly, acting as a cytoprotective agent. Finally, when its job is done, it's metabolized in the liver by the enzyme CYP1A2 into 6-hydroxymelatonin, which is then excreted.
And for our final atypical messenger, we turn to a molecule you definitely know, but in a totally different context. ATP, adenosine triphosphate. Yep, we all learned about it in high school biology as the fundamental energy currency of the cell. Well, it turns out the brain co-opted this universal energy molecule to serve as a critical neurotransmitter.
This diagram shows the whole purinergic ecosystem. ATP can be co-packaged into synaptic vesicles right alongside other neurotransmitters, using a specific transporter called VNUT. Once released, it can act on its own receptors. But it doesn't stay as ATP for long. Out in the synapse, a team of enzymes called ectonucleotidases quickly snips it down, first to ADP, then to AMP, and finally to adenosine. And adenosine is a super important signaling molecule in its own right, often acting as an inhibitory, calming signal. Then, to complete the cycle, adenosine is brought back inside the cell by transporters and can be recycled back into ATP by an enzyme called adenosine kinase. Ready for the next round.
Now, the receptors for this system fall into 2 main families. The P1 family receptors all bind to adenosine, and they're all G-protein coupled, which means they trigger slower, modulatory effects inside the cell. The P2 family receptors, they bind to ATP and ADP, and this family is split into 2 distinct types. You've got the P2X receptors, which are fast-acting ligand-gated ion channels, and the P2Y receptors, which, like the P1 family, are G-protein coupled.
Let's just zoom in on those adenosine receptors because they have major clinical relevance. The A1 receptor, which inhibits the enzyme adenylyl cyclase, is a therapeutic target for epilepsy since adenosine acts as a natural anticonvulsant. In contrast, the A2A receptor stimulates that same enzyme, and it's a key target in treating Parkinson's disease. This system is also implicated in some rare genetic disorders. For instance, Lesch-Nyhan syndrome is a devastating condition caused by a mutation in the HGPRT enzyme, which is involved in purine salvage. And mutations in adenosine deaminase, which breaks down adenosine, can lead to severe sleep disorders. And this really brings us full circle. From messengers that talk backward to those made of gas, from proteins shipped from the nucleus to the cell's own energy currency pulling double duty, these so-called atypical neurotransmitters show us that the brain's communication system is far more creative and flexible than any simple rulebook could ever capture. They remind us that in neuroscience, sometimes the exceptions are what reveal the true elegance of the rules.